Hello, everyone, and welcome to today's webinar, Sepsis, Diagnostic Perspectives for Clinical Microbiology Laboratories. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BD. To learn more, visit bd.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Kenneth S. Thomson, Clinical Professor of Microbiology and Immunology, University of Louisville School of Medicine. Dr. Thomson, you may now begin your presentation.
Thank you, Marie. I feel privileged to have been invited to participate in this World Sepsis Day presentation. My aim is to briefly review the incidence and mortality of sepsis, and then move on to the detection challenges facing microbiology laboratories. The focus will be on concepts, perspectives, and priorities, rather than a detailed technical guide of how to perform every test and provide a laboratory service, or whether a company A's test or device is better than that of company B. Here is my disclosure. Basics. This and the other initial slides give a perspective of the sepsis problem. Sepsis and septicemia are not the same thing. Septicemia is a non-specific term describing the condition of a person who is unwell and has a bloodstream infection. Sepsis goes beyond this. It is diagnosed clinically by the presence of acute infection and new organ dysfunction. This term, as indicated by the definition in the slide, focuses on the host response as the major source of morbidity and mortality. It only requires that infection be suspected, not proven. This is to cover the reality that many cases do not have confirmation of the infection, even though the chain of events leading to death is presumed to be triggered by bacterial, fungal, viral, or parasitic pathogens. There can be many causes of sepsis, but the leading cause is bloodstream infections. This slide provides various estimates from the first single study spanning the globe, as opposed to other estimates that are based on data from multiple studies. This was a 27-year study extending to 195 countries and territories. It reports figures that are more than double previous global estimates, probably because it includes more data from previously underrepresented low-income and middle-income countries in which sepsis incidence and mortality are considerably higher. Annually, it is estimated that there are approximately 50 million cases of sepsis, approximately 11 million deaths, and sepsis causes approximately 20% of all global deaths. The encouraging news is that incidence is decreasing around 1% annually in nearly every location worldwide. In the US, sepsis is the most common cause of hospital deaths at an annual cost that exceeds $24 billion. Having looked at the estimated global burden of sepsis, the question is, how accurate are the data? Firstly, most data come from hospitals in high-income countries and are not representative of low-income countries. Secondly, they are of variable quality. Some are from electronic health records and others are from death certificates. Some hospital administrative databases are potentially inaccurate and may be based on non-standardized definitions of sepsis that are used for case reports and death certificates. In short, estimates rely on disparate data. This hampers comparability over time or by location or even within the same population. Also, most studies are restricted to patients admitted to hospital and exclude children, the underlying cause of illness, and assess data for only 1 or a few years. Then there are multiple possible sepsis-related classification codes. Bottom line is that estimates of sepsis burden vary in quality and accuracy. Before leaving the issue of estimates, there are a couple of quantitative analytical tools for comparing data from different populations. These are the Socio-Demographic Index, or SDI, which is the term I will use, and the Healthcare Access Quality Index, or HAQ. The SDI is something to consider when comparing populations. It ranks a population's spectrum of development. This is based on things that can be measured: people's incomes, their level of education, and how successfully they breed. Here's what the SDI analysis tells us. The highest mortality occurs in areas with the lowest SDI, and these low SDI locations have the highest rate of sepsis-related deaths due to infection. High SDI locations have the highest rate of sepsis-related deaths due to non-communicable diseases. The quality of the data: low and middle income countries are less likely to have high quality data sources concerning causes of death or hospitalization or sepsis coding. Healthcare access. The HAQ is based on 32 diseases that would not be fatal if there was an effective healthcare infrastructure. A population's access to good healthcare is calculated to assign a score of 0 to 100 for each location. What the HAQ tells us is that the highest burden of sepsis is in locations that have the least capability to prevent, identify, or treat sepsis. It certainly is nice to be able to measure this as a number, but I really don't know how much time and effort goes into it. I'm sure the HAQ is a nice term for infectious disease professionals, especially those employed in academia, to be exposed to once in their lifetimes. I could be wrong, but I think that with much less time and effort, you could probably get enough useful information about healthcare access and quality from a 20-minute chat with a couple of local general practitioners. Some information about underlying causes of sepsis. Diarrheal disease is the most common of all underlying causes of sepsis cases, but not necessarily mortality. And road traffic injury is the most common underlying causative injury. Maternal disorders are the most common non-communicable disease that is complicated by sepsis, in particular causing septic appendicitis. There are 2 age groups for peak incidence for both cases and deaths. These are children younger than 5 years with diarrhea and older adults. When it comes to mortality, the most common underlying cause of mortality is lower respiratory infection. Treatment of sepsis is a matter of great urgency. Blood is collected as soon as possible followed by the initiation of empirical, that is, best guess antibiotic therapy. Typically, broad-spectrum antibiotics are used because the pathogen is unknown at this time, and there are no susceptibility results available to guide appropriate therapy. The choice of empirical therapy is usually based on the local antibiogram or recommended guidelines, such as those of the Infectious Diseases Society of America, the IDSA. This therapy may include antibiotics such as amikacin, carbapenem, piperacillin tazobactam, cefepime, or vancomycin. Unfortunately, the pathogen may be very resistant and not covered by empirical therapy, in which case there is a greater risk of death. A patient with community onset sepsis, that is a positive blood culture within 2 days of admission, is less likely to have a resistant pathogen. The community onset sepsis patients, about 1 in 8, have a resistant gram-positive pathogen, usually MRSA, rarely VRE, and about 1 in 8 will have a resistant gram-negative pathogen, mostly a ceftriaxone-resistant organism, which will rarely be an ESBL or carbapenemase producer. That is, the need for broad-spectrum antibiotics is less for community onset patients. Giving such antibiotics to patients who don't need them enhances broad-spectrum antibiotic resistance selection, and that is an increasing concern. Whether it is hospital-acquired or community onset sepsis, the infection must be treated correctly and quickly to prevent death of the patient and the emergence of resistance. What is needed is better tools to predict patients at greatest risk of a multi-drug resistant pathogen infection and to provide an accurate guide to directed therapy. So let's look at how good empirical therapy is. This slide is from a multi-center comparison of outcomes for inappropriate and appropriate empirical therapy for bloodstream infections in patients with febrile neutropenia. Not all patients in the study had sepsis, but the findings are relevant and instructive for today's topic. Results were assessed according to adherence to prescribing to IDSA empirical therapy guidelines, and if the guidelines led to appropriate therapy for each patient's infection. Compliance with the guidelines was achieved in 87% of 1,615 bloodstream infections. Even with such good compliance, 24% of patients received inappropriate, that is, ineffective empirical therapy. That is, following guideline recommendations did not ensure that patients received effective antibiotics for their infections. As antibiotic resistance increases, ineffective empirical therapy is going to be an increasing problem. This slide shows data for 5 organisms. Not surprisingly, empirical therapy did best for E. coli infections, being appropriate 82% of the time. It was inappropriate in 18% of cases. That is, almost 1 in every 5 patients, for reasons such as ESBL production or multi-drug resistance, had inappropriate therapy. Looking at the second organism listed, Pseudomonas aeruginosa, there were 251 blood culture isolates, 71 of which were multi-drug resistant. For 51% of the multi-drug resistant Pseudomonas aeruginosa, the patient received inappropriate therapy. This is a whopping level of inappropriate empirical therapy, especially since Pseudomonas aeruginosa is noted for being associated with high rates of sepsis and death among neutropenic patients. The other organisms in the slide are there for your information only and do not add to the points already made. I won't discuss them, but it may be pertinent information for you to have later. When the study data are analysed for 30-day mortality, multi-drug resistance and the organism type were important. Inappropriate empirical therapy was associated with increased mortality in gram-negative bloodstream infections, but not so in gram-positive bloodstream infections. There were slightly more deaths with inappropriate therapy for gram-positives, but the difference was not significant. In contrast, inappropriate therapy was associated with greater mortality in gram-negative bloodstream infections, and especially in infections caused by Pseudomonas aeruginosa. Independent risk factors for Pseudomonas aeruginosa associated mortality was septic shock, pneumonia, and inappropriate empirical therapy. If you look at the table, even with appropriate empirical therapy, Pseudomonas was associated with higher mortality than the other organisms. Major points from this study were that bloodstream infections by multi-drug resistant isolates were common. In spite of IDSA guidelines, patients frequently received inappropriate empirical therapy. And finally, Pseudomonas aeruginosa bloodstream infections are very bad. Detection of Pseudomonas aeruginosa in a blood culture has high prognostic importance. For these reasons, it is vital that accurate susceptibility results are reported as quickly as possible. This is a big challenge for the microbiology laboratory. When the laboratory investigates suspected sepsis, because of the high risk of death, it is absolutely essential to provide the best quality of service. Let's now look at the essential diagnostic perspectives. The first perspective is diagnostic stewardship. This term is integral to the management of sepsis. In the future, tests should be tailored to the treatment needs of individual patients. We cannot do this yet, but our knowledge of sepsis and diagnostics is experiencing explosive growth with advances in computational power, access to cloud services, new bioinformatics tools, and new technologies emerging. All of this will lead to dramatic improvements in rapid laboratory testing. For the present, however, the selection of appropriate tests is done on a patient population basis rather than the needs of the individual patient, and blood culture remains the single standard procedure for diagnosis of bloodstream infections. I strongly believe that the detection of bloodstream infection is one of the most important functions of the clinical laboratory. Therefore, a laboratory needs to use the best available blood culture system. You may be able to cut costs on other tests like urine cultures, but always make sure you have the best quality blood culture system you can get. Some basics about providing a blood culture service. Speed and accuracy are essential. The shorter the turnaround time, the greater the chance of saving a patient's life. If a blood culture comes to the lab, it must be processed immediately. When staff start their shift and there are positive blood cultures, and growth from cultures of other less urgent specimens, the blood cultures are the immediate priority. If there is something to report from a positive blood culture, report it as soon as possible. Conventional microbiology can take 3 days to yield identification and susceptibility results for directed therapy. This is way too slow. Each hour of delay in getting the patient onto effective therapy increases the risk of a fatal outcome. The aim is to reduce the time to initiate appropriate directed therapy. This is the period of maximum vulnerability for the patient. Accurate, rapid tests are essential. Optimum turnaround time is critical. Eliminate things that may cause delays or impair quality. It is important to monitor quality indicators, such as turnaround times for reporting gram stains and molecular tests. There are many quality indicators that are important to monitor. Otherwise, quality might be sliding unnoticed to the detriment of patients. Here are the essentials for the laboratory workup. Process samples immediately on arrival. Positive cultures must be reported immediately. It is vital that cultures are followed up 24 hours a day, 7 days a week. Is the big one. It is essential to have dedicated microbiologists to process positive bloods without delay. They must be able to do gram stain reports and initiate and interpret follow-up tests such as identifications, antibiotic susceptibilities, resistant mechanism tests for ESBLs, carbapenemase-producing organisms, also known as CPOs, detection of the MEK gene, and so on. Sometimes the question arises whether to process blood cultures in-house or send them to a central laboratory. Processing in-house is ideal, but you must have every one of the essentials at the top of this slide. Otherwise, use a central laboratory to provide these essential requirements. If you use a central laboratory service, it is only as good as the courier service. Deliveries every 2 to 4 hours for 24 hours each day would be good. This is something that needs quality indicator monitoring. Why do blood cultures? The physician wants to know the answers to 2 questions. Does my patient have a bloodstream infection? And if so, what do I treat with? This information is wanted quickly. If the blood culture is positive, the gram stain must be quickly done and reported. Microscopy can indicate if the pathogen is bacterial or fungal, and possibly what the organism may be. This can give the physician a clue for therapy. Ideally, the gram stain results should be followed by a direct organism identification from the positive blood culture by molecular or moldy testing. Early identification information can be helpful to support or modify the choice of empirical therapy prior to susceptibility results being available. As we are aware, empirical therapy is not always the correct therapy, so the need for rapid, accurate susceptibility information is critical. If direct identifications from the blood culture are not possible, there will be a delay until colonies from the subcultures are available to test, and then a further delay for however long the identification test takes. Okay, here's one for the advanced players. Gram stain of gram-negative rods in a blood culture. Notice the narrowness of some of the cells, particularly in the center of the slide. This is suggestive of the organism possibly being Pseudomonas aeruginosa. Slides like this show why it is absolutely vital to have a competent microbiologist doing a gram stain interpretation for a positive blood culture. The gram stain provides important information for the choice of therapy. On my very best day at the bench, it would have been a stretch for me to have made the Pseudomonas call on this one. But we do have some superb microbiologists in this country who can make calls like this. To me, it is a scandal that contributions like interpreting this sort of gram stain as Pseudomonas are not recognised when it's time for annual evaluations and pay raises. And incidentally, I'm confident that my wife Gina could look at this slide from 20 yards away and say, that's Pseudomonas. Here is another one for the advanced players. Acinetobacter is gram-negative, yet in this positive blood culture, it is appearing as gram-positive cocci. This is a distinctive feature of Pseudomonas that is sometimes seen and can be recognized by trained microbiologists, but not by non-microbiologists. The point in showing these 2 gram stains is to drive home that it is critical to have proper microbiologists to do follow-up tests and interpretations. Here are some representative examples of molecular tests that can be done on positive blood cultures. They take about 90 minutes, which is rapid, and they have high specificity. Positive molecular results can be very helpful. Detecting resistance mechanisms can eliminate consideration of certain drugs and possibly help get the patient on effective therapy many hours sooner than otherwise. This can significantly reduce length of stay, save a life, improve antibiotic stewardship, and save money. The downside of current molecular tests is their limited target ranges, and while detection of resistance genes can reduce the possibility of the patient receiving the wrong drug, it is not the same as a susceptibility test, which can indicate the drugs to which the pathogen is susceptible as well as resistant. Alan Junkins at Louisville's Norton Hospital had an interesting experience with Verigene testing on positive blood cultures. Positive bottle from a pediatric patient indicated a bloodstream infection caused by KPC producing E. coli. Yet in the susceptibility test, the isolate was very susceptible and gave no hint to initiate a carbapenemase test. Without the molecular test, a finding of high significance would have been missed. Alan has seen this more than once, and from posts on ClinMicroNet, other laboratories have too. In Alan's case, it was not a Verigene false positive result. At our lab, we confirmed carbapenemase production by detecting carbapenem hydrolysis, and also saw a significant increase in carbapenem MICs when tested with 100-fold higher than standard inoculum, an inoculum that might occur in a patient's infection. Another important point about detecting this isolate as a carbapenemase producer is that KPC is a transmissible carbapenemase, and therefore the isolate could have been a source of KPC transmission. The bottom line with molecular tests of positive blood cultures is that they are definitely worth doing, as positive results can save lives. This slide reinforces the point that negative molecular tests can be falsely negative, which is important to remember. The table data are from a study in which the Cepheid and Verigene assays were used to test 250 well-characterized carbapenemase producers. The Cepheid PCR targets are the carbapenemase families referred to as the Big Five: NDM, KPC, IMP, VIM, and OXA48. The Verigene microarray has more targets, but requires an organism identification to detect them. The limited ranges of the two tests meant that both were incapable of detecting many carbapenemase encoding genes. Carbapenomases shown in blue are not targets for these tests. Therefore, both tests missed genes encoding SME, IMI, NMCA, GIM, and SPM carbapenemases. In addition, Cepheid cannot detect genes for the oxocarbapenemases of Acinetobacter baumannii. Among its target genes, Cepheid had problems with the IMP family. It could not detect genes encoding IMP7, IMP8, IMP13, IMP14, IMP18, and IMP27. To me, this problem indicates that its target range is not the Big Five, but more likely should be called the Big 4.5. Veregene did not detect KPC or NDM genes of Morganella, Serratia, Klebsiella variocola, Hafnia, Providencia, and Salmonella because it could not identify these organisms. These findings are just for carbapenemase gene detection. Molecular tests can also miss genes for ESBLs and AMPCs. So summing up all this, molecular tests are helpful if you get a positive result. Unfortunately, their target ranges are not keeping up with what's happening with antimicrobial resistance. They are not standalone tests. Before moving on to other tests performed on positive blood cultures, what about bypassing blood cultures altogether and using molecular tests directly on whole blood? Doing molecular tests directly on whole blood would eliminate the delay of waiting for a blood culture to go positive. This is theoretically feasible with current multiplex PCR tests for target pathogens and resistant genes. Advantages could be that unlike blood cultures, antibiotics don't interfere with these tests, and they have the potential to be helpful if blood cultures are negative. But, unfortunately, these tests are less sensitive than current multiplex PCR tests on positive blood cultures, and this makes them unreliable. T2 magnetic resonance is more sensitive than multiplex PCR. This is pathogen-specific PCR with miniaturized magnetic resonance. After the PCR amplification step, the amplified DNA is detected by hybridization to probe-enriched superparamagnetic nanoparticles, which causes large changes in the T2 signal and confers superior sensitivity. This technology offers great potential, but currently there is the disadvantage of limited target ranges. T2 can test for only 5 species of Candida and not including the very important spreading treatment-resistant Candida auris. Similarly, it can only test for 5 bacterial species, and this approach does not include antimicrobial resistance targets. The omics technologies have great potential for the future. In-depth studies of the classes of biochemicals listed in this slide will eventually help us to understand the interplay between host and pathogen and improve sepsis recognition and appropriateness of antimicrobial therapy on an individual patient basis. At this time, there remain many technical challenges to resolve. As one example, in transcriptomics, the analysis of RNA transcripts, dynamic changes in transcription occur that result in a potential lack of reproducibility and lack of understanding of the results in certain patient populations, such as those with neutropenia or solid organ transplants. The sensitivity and specificity of these types of technologies have not been sufficiently studied to use them as standalone tests. And they require great investment in specialized equipment and high levels of technical expertise to analyze and interpret data. They also require standardization of methods and bioinformatics tools. Finally, going back to testing positive blood cultures, let's look at some growth-based technologies, but not the slow conventional tests that were in use 50 years ago and are still being used today. This slide shows two rapid technologies that can be performed directly on positive blood cultures: the Accelerate Pheno and the UCAST RAST method. RAST is the abbreviation for Rapid Antimicrobial Susceptibility Test. Briefly, the Pheno is an identification antibiotic susceptibility instrument. Identifications can be done by FISH technology in approximately 2 hours or with a laboratory's own system. To me, the susceptibility test is what will determine the future of the Pheno. It is based on novel technology in which cell growth is monitored by powerful imaging technology that compares the morphology of bacterial cells in the presence and absence of antimicrobials. MICs are obtained in approximately 7 hours. This is a neat concept, and the Pheno is now in a number of clinical laboratories. Some laboratories use it as a reliable standalone test and report results without reservation. Other laboratories do not have the same comfort level and back up Pheno results with conventional tests, sometimes finding disagreement between the Pheno and the conventional results. This raises the issue of whether early antibiotic susceptibility results are okay to report for serious infections if there is uncertainty about their accuracy. No susceptibility test is perfect, but some tests are unacceptably error-prone. So the thing to determine is if the accuracy of the Pheno is acceptable for your laboratory. The UCAST RAST method is a rapid disc diffusion susceptibility test in which the susceptibility plate is inoculated with 100 to 150 microliters of positive blood culture broth. If there is sufficient growth to measure inhibition zones, the plates can be read after 4 hours. If more growth is needed, zones can be read at 6 hours or even as late as 8 hours. There are organism-specific interpretive criteria for each reading time, and readings must be made within a 10-minute time frame, or else it is necessary to wait for the next reading time. The test has been validated for 18 drugs against the organisms listed in the slide to cover most commonly encountered bloodstream infections. It's cheap, easily adapted as new drugs and organisms are added in its range, and the UCAST people are working hard and fast at validating additional drugs and pathogens. Unlike molecular tests, there is no special equipment requirement, but for a laboratory with a total laboratory automation system, such as the BD Keystra or Copan WASP, RAST has the potential for automated testing and reading. There are already papers starting to come out about this. There are some limitations. When we looked at it a bit over a year ago, we found that some of the drug discs were not available in the United States at the required potencies. Also, the need to measure zones within the prescribed 10-minute periods must be complied with. Overall, the potential of this test is impressive. It provides the rapid and accurate detection of resistance and susceptibility that is urgently needed for the main bloodstream pathogens. This test is definitely worth considering as an addition to the workup performed directly on a positive blood culture. In conclusion, because blood culture remains the single standard procedure for diagnosis of bloodstream infections and is likely to be for some time, what are some targets to aim for to reduce turnaround times and improve quality? One target is shortening the time to blood culture positivity. In theory, there is considerable scope for this. Most blood cultures incubate for at least 10 hours before growth is detected. This time can be reduced if the positive signal can be amplified to make it stronger and therefore detectable earlier. Other ways to achieve faster detection would be to enhance the sensitivity of the detector of the blood culture system or find a better target and detection system. Under optimal conditions, bacteria multiply about every 20 to 40 minutes. Every time they divide, they produce a positive signal. The problem is it is a weak signal that gets stronger with each generation of growth, but it currently can't be detected until 10 or more hours of growth. But the positive signal is there to be detected right from the first hour or so. What we need is better detection technology to detect positivity earlier. This should be an achievable target. The next ones are targets your laboratory may have already achieved, but if not, they are ones to aim for. Gram stains should be done immediately and reported without delay. Identifications are best initiated immediately rather than waiting for colonies to grow from subcultures. Molecular tests, yes, do them on positive blood cultures. Antimicrobial resistance is an important driver of sepsis, especially in healthcare settings. Rapid susceptibility results are vital. The UCAST RAST method can make this possible. Also, combination therapy is often needed in this age of increasing resistance, but we don't have a convenient, accurate susceptibility test to guide combination therapy. The time has come for such testing, especially for gram negatives. I strongly believe that all of these are essential targets to aim for to succeed in one of the most important functions of the Clinical Microbiology Laboratory, the investigation of suspected sepsis. On that note, I thank you for your attention and also thank Gina Thomson at the University of Louisville Hospital and Kevin McNabb at New Hanover Regional Medical Center in Wilmington, North Carolina for their help with the preparation of this presentation.
Thank you, Dr. Thomson, for your informative presentation. We'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, what are MDR pathogens?
MDR stands for multi-drug resistance. There are many classifications of multi-drug resistance, extreme drug resistance. I go by the CDC classification. Multi-drug resistance is resistance to at least one agent from 3 different antibiotic classes.
Okay, thank you. Our next question is, do we need better guidelines for empirical therapy?
Guidelines need updating to respond to constantly changing antimicrobial resistance and availability of new antimicrobials. The current IDSA guideline for the bloodstream study I presented was first published in 1997, updated in 2002, and then updated again in 2010. This update was mainly to provide clearer definitions of levels of risk for patients. It did mention the need for KPC coverage if the patient had previously been exposed to the organism or the patient's hospital had a high rate of endemicity. Then the recommendation for appropriate empirical therapy was to consider early use of polymyxin or tigecycline, antibiotics today that should be considered inappropriate.
Okay.
The recommendation in that guideline for high-risk patients is monotherapy with an anti-pseudomonal beta-lactam agent such as cefepime, carbapenem, or piptazo, with other agents to be added only if there are complications such as hypotension or pneumonia, or if antimicrobial resistance is suspected or proven. Vancomycin and other anti-gram-positive agents are not recommended as part of the initial antibiotic regimen. That's what it was in 2010, and that's what was used in that study that was published. I showed you that was published in 2020. So much has changed now. There are other carbophenamases besides KPCs. There are new agents such as ceftolozane, tazobactam, new beta-lactamase inhibitor combinations. Fungal infections are more common. So yes, I think we need better guidelines for empirical therapy.
Okay, thank you. Next question. Does CLSI have a RAST method for blood cultures?
Does CLSI have a RAST method for blood cultures? Well, sort of. Unlike the UCAST test, which has readings at 4, 6, and 8 hours, the CLSI version must incubate for much longer, for 16 to 18 hours, just like a conventional disk diffusion test. And it's only applied to Enterobacteriales, whereas the UCAST version is specific for 2 Enterobacteriales, E. coli and Klebsiella, plus Pseudomonas aeruginosa, Acinetobacter, Staph aureus, Strep pneumoniae, and 2 species of Enterococcus. Also, the CLSI version is only for 6 antibiotics, whereas the UCAST RAST test is for 18 antibiotics. So UCAST is much more rapid and covers a wider range of pathogens and drugs. To me, the short incubation time for the UCAST test versus the much longer incubation for the CLSI test indicates very different mindsets, with the UCAST priority being clinical relevance and the CLSI priority being reproducibility. It seems crystal clear to me which is the better test in the light of risk of mortality in sepsis increasing every 1 hour of delay.
All right. Our next question, is there a rule said that each site on the body has specific pathogens?
Could you say that again, please?
Yes. Is there a rule said that each site on the body has specific pathogens?
Not that I'm aware of. And if there was a rule, I'm not sure the bacteria would behave and stick to it. Neisseria gonorrhea is more specific about where you find it. Most pathogens can occur anywhere.
Okay, thank you. Our next question, based on the methods you listed for blood culture evaluation for sepsis, which method do you preferably recommend and why?
I I said at the start of the presentation, I wasn't going to talk about one method being better than another, comparing different products. I guess it varies from lab to lab what's going to be the best system, but I'm not going to make a recommendation.
Okay, thank you. Our next question, could you provide more details about the UCAS RAS test and Accelerate Pheno test?
More details. I think the Pheno is a neat concept. To me, we looked at it a couple of years ago, and I didn't think it was ready for prime time because we had consistent problems with certain antibiotics. I think ceftazidime and cefepime might have been ones that we didn't get answers that agreed with microdilution. The RAS test, more specific information. It's pretty simple. We just have to be able to read a susceptibility test by measuring the zone diameter. That is an art that's not as strong these days as it used to be, and you may have to train people how to read zones more accurately. But I think it's a beautiful test. We actually used it on a total laboratory automation system. And what that meant was that a positive blood culture could be put up immediately, and then photos of the zones were taken at exactly 4 hours, 6 hours, and 8 hours, so that you didn't miss the reading times, and the photos were there for experienced microbiologists to check, to be used for teaching purposes. That was very handy. I think we got something like 74% of interpretations within 6 hours, 100% of interpretations within 8 hours. I liked it very much. I thought it was great and could save lives. But it was very accurate. The only one we had problems with, I think, was erythromycin. Clindamycin, we missed inducible resistance sometimes with Staph aureus. But for most of the other tests, it was great. It was irritating that we couldn't get all the drugs that we wanted at the right potencies. When we did the study, cefotaxime and ceftazidime were two of those drugs. I hope they're available now. I like it. I think it's worth doing. If you get an early guide to susceptibility, what drug to have the patient on, that can save lives. I think it's still developing this method, but it's got great potential. Very excited about it.
Okay, thank you. Our next question, how can the mortality rate in sepsis be prevented?
A lot of people die of sepsis. I think we can reduce mortality. The rate's going down about 1% every year all over the place. I think that's because of improved blood culture methods. Getting an earlier guide to what drugs to treat with, that shortens the time that the patient is vulnerable. Generally good infectious disease practices like reducing communicable diseases, especially diarrhea, things like that are important. Good infection control to stop the spread of multi-drug resistance pathogens in closed communities like in medical institutions. Lots of things, but they're some of the main ones. And just make sure that you don't have any glitches in your workup of blood cultures. And make sure that when a doctor issues a request for a blood culture, that's acted on quickly, the sample gets to the lab really quickly, it's processed very quickly when it gets to the lab, and all the other things I mentioned, like interpreting and reporting gram stains really quickly, just cut down on delays. There's lots of quality indicators that you can pay attention to, and all of those are going to be helpful in reducing the mortality.
All right, it looks like we have time for one more question. I really appreciate the presentation. It was quite informative. Kindly share the presentation slides.
Share the slides?
Yeah,
I'd be happy to do that. I think Laboratories make them available, don't they?
Yes. Yes, Laboratories will send out an email to everyone that has attended or signs up, and they will get access to the slides. They will also be on demand. My apologies. Let's do one more question. Our hospital has a surge of COVID patients. Blood cultures are routine for each patient admitted. Does the virus survive in blood culture bottle broths?
That's a really good question for another reason. I don't know, someone else could tell you. I haven't done any tests like that, but COVID in a hospital, what that does is you're going to have patients in ICU for longer periods than previously, lengthy stays in ICU, ventilated. You've got short staffing. You're going to have breakdowns in infection control. People are overworked. And what that means is that there is increased risk of patients getting candidemia. And that's something that you really need to look out for and get onto very quickly. So COVID might be changing things in ways that we haven't really appreciated yet. But I'd be looking out for candida bloodstream infections. They may go up as the staff get worn down and the patients stay in there for long periods.
All right. Well, thank you again, Dr. Thomson, for your time today and your important research. We would also like to thank Laboratories and our sponsor, BD, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.